Cancer is the change of what was once upon a time a normal cell into a cell which grows and proliferates in an uncontrolled fashion. Every normal cell has a blueprint, a DNA code that normally tells it how to behave, and if that DNA code or blueprint becomes changed or mutated, then that can lead to genes running amok, and it can lead to the cell growing in a completely uncontrolled fashion, and that is in essence a cancer cell. Malignant melanoma is a special type of skin cancer. We all have within our skin cells called melanocytes, and these are cells that normally produce pigment or colour. For example, when we go into the sun, sunlight is harmful to our skin, so normally these melanocytes will produce a brown pigment, what we call a tan, which helps protect us from this ultraviolet, this radiation. And unfortunately, if you're exposed to too much sunlight or ultraviolet radiation, then these melanocytes can accumulate mutations or damage in DNA. That means the melanocyte, the normal melanocyte, becomes a cancer cell. When it becomes a cancer cell, we call it a melanoma. Some patients who get melanoma on their skin can have it successfully excised by a surgeon and it will never come back. But in quite a significant number of patients, even when it's excised, it comes back a couple of years later. And when melanomas come back, they are invariably fatal. Up until about 2 years ago, we would have treated a melanoma patient with largely surgery if the cancer was only on the skin, and if the cancer had spread, we would have used chemotherapy, but only about 10% of patients responded. What we discovered at the Sanger Institute about 5 or 6 years ago was that in about half of the cases of melanoma skin cancer, the melanoma is essentially harbouring a mutation or DNA mistake in a very specific gene, and that gene is called BRAF. And having that mutation in BRAF essentially gives the melanoma cancer cell constant acceleration. Scientists found that BRAF was significant in melanoma by essentially sequencing large numbers of these cancers using technology that's been developed over the last 10 years. And in essence, when you sequence a large number of any cancer, you can simply ask what are the most frequent abnormalities or mutations we see, and that invariably tells you what's important to the cancer. And in this case, in melanoma, we found that up to half of melanoma had a mutation in the gene BRAF. These particular mutations in melanoma are its Achilles heel, and if we use a new drug, a brand new treatment, in these patients, the response rate goes from 10% to 70%.